bio majors here? How many of you have had introductory biology at a university level? Oh, okay, all right. So some of this should be reviewed, although I'm not sure how much you remember from introductory biology. I know what our bio students remember, and often it's not what we think it is. But what I'm going to do today is go through some of, as Tanajit said, some of the big data issues associated with specifically RNA-seq experiments. I'll explain what RNA-seq is, why it is really, really big data and becoming bigger literally by the minute in modern biology and medicine, and then some of the issues associated with the interpretation of this data. What are the problems? What needs to be solved in this field? And I should mention that I am not a statistician, I am not a mathematician, I am a molecular geneticist, and we are actually collecting this data on campus. So the major part of my job description, in addition to teaching intro bio every other year, and developmental biology and some bioinformatics, is actually collecting data like this and making sure the experiments work. So I'm a wet lab biologist, but as every wet lab biologist in the 21st century is finding out, they wished, and this is the case with me, I had a triple major in biology, comp sci, and math. But we have to make do with collaborations because that's obviously a lot of work to do as an undergraduate. So just a little bit before I go into what RNA-seq is on the significance of this. So what this is doing, big picture here, is revealing, telling the investigator or the physician which genes are actually being activated, which ones are expressed. Everybody's heard about sequencing the human genome and all of the other thousands of genomes out there that have been sequenced. So that is basically determining the letters, the nucleotides on the DNA level. Important, it gives us ideas if there's a mutation in a gene that is associated with a disease, but it's not telling us which genes are actually activated in a given cell. And that is what is actually causing your brain cell to be different than a skin cell. It's not at the DNA level, it's at the RNA level, and this is what RNA-seq does. It also distinguishes whether a cell is going to become cancerous or not cancerous, healthy or not healthy. So this is so important now in modern medicine. Just a few very recent titles. I just read an article entitled Whole Genome Analysis. When each patient, we're not talking about all of them, when each patient is a big data problem, the analysis here. Physicians are certainly not trained in big data, but they're going to need to be in the 21st century here. And just this morning, NIH sent out a call for proposals across all of NIH, kind of a trans-NIH, the diabetes, the metabolism, the development, all these different sections of the National Institutes of Health talked about calling for proposals to deal with this problem. And a quote, spectacular opportunities for all you math majors out there interested in big data. Really spectacular opportunities and immense challenges focusing on the biomedicine. So this topic is highly relevant. So just a little bit of review in case you don't remember this or have not had it in intro bio to look at the levels at which we're talking here. Simplistically, the central dogma of molecular biology since the days of Watson and Crick. You begin with DNA, which is used as a template for RNA, which is then used as a template to make protein in every cell on the planet. That was literally a dogma. It's still called a dogma, although two problems with this. One is we know there's an exception to it. Unlike in physics, where there are very few exceptions to the laws or the rules or the dogmas, in biology, there is an exception to virtually every single thing that we say. Every sentence you'll read in a textbook that generalizes, there'll be an exception, which makes data analysis difficult. So the one problem is we now know that there are viruses that have as their genome, like the HIV virus, RNA that goes to DNA and then back to RNA again. So a little bit of a twist in this central dogma. That's the first issue. There are exceptions. The second one is that it's way more complex in eukaryotic, that is our types of cells here, as opposed to bacterial cells. But even in bacterial prokaryotic cells, this is way too simplistic. So for what we'll be looking at for RNA-seq, this is a typical eukaryotic cell. So here is the nucleus, here is the cytoplasm. You're going to begin with the DNA, and this is basically the same in every cell in your body, with the exception of immune cells, where there are rearrangements at the DNA level. But the DNA in your brain is the same as the DNA in your skin. What makes those different? It's the RNA. So you have transcription of the DNA, so this is the process, into what we call a pre-RNA. It's not ready to be used as a template for protein synthesis yet. We have these things called introns. Nobody completely understands the evolutionary advantage of having for a gene, the vast majority of that gene be chopped out, just completely gotten rid of. This is called splicing. And then the pieces that are left are spliced together to form what we call the messenger RNA, mRNA. That mRNA is then exported, sometimes not. This is a highly regulated procedure here. So sometimes it's exported through these nuclear pores into the cytoplasm. Then it gets translated into a protein product. And what I want to pay special attention to here, because this is a really major problem with RNA-seq, is this splicing. So if you look at the top there, at the level of the DNA, you'll see exon 1, 2, 3, 4, 5. These are the parts of the gene that will be transcribed along with these introns in the pre-RNA. So this is the pre-mRNA here. Everything gets transcribed. And then, as I said a minute ago, these introns are spliced out. But it's not that simple. Sometimes you have all of the introns spliced out, and all of the exons are spliced back together. And you get this mRNA. In other situations, same gene being expressed in a different cell type will have what we call alternative splicing and only have exons 1, 2, 4, and 5. In a different cell type, same gene will have 1, 2, 3, and 5. There are genes in our genome that have up to 50 to 60 different exons, and you could do the math on this, combinatorial, the different combinations that you could get from 60 different exons. So you have one gene, and each of those different protein products does something different in the cell. So this in itself really causes problems for what we're going to be talking about, the RNA-seq. And until the advent of RNA-seq, we had no idea how many of these different, what we call splice variants, actually exist. So kind of keep this in mind as we discuss later the RNA-seq. So I know you've been discussing all term, and I'm not going to read this. What is big data? By any definition, and we'll look at a few numbers here, this is big data. I just put a few of the definitions that biologists are kind of interested in using, and really, this is kind of old, but it really stresses this idea of the volume, just the sheer amount of data, but it's not only that with biology. Certainly how quickly it is being generated, and the different types. This emphasis on the different types of data and how you bring them together when they're very different, and the complexity of that data. But then in biology, we have this growing problem of veracity. It's not that people are making up data, but how good is that data? What confidence do you have in sequence coming out of the actual instrument? So basically, for us, it's a combination of sheer volume, complexity, the speed that our algorithms are not keeping up with the analysis. We don't understand even how to analyze these data, and this uncertainty level, the complexity and the uncertainty. This is just looking at sequencing, and this is it doesn't even go to 2013. It would be way up here by now. Way back, and we're talking about only a few years ago, when every biologist in the biology department was actually trained in doing biology, we were gathering sequence data at a minimal level. What they call it here in this kind of demeaning way, a few flurries of sequence. And of course, I did my whole PhD dissertation right around here with these flurries of sequence. That was a PhD, sequencing a gene. Now, with what we call next-gen sequencing, this is rising, more than exponentially, of course. Just incredible amounts of data, and the technology is improving literally every month. We get 150 gigabytes, and this is just the finished sequence of a human genome or transcriptome. And by the way, humans have about, anybody know how many genes? Pardon me?
46. No, 20. No.
Chromosomes.
Oh, that's right.
Yeah, chromosomes. And those are the physical locations upon which the DNA covered in all this protein actually reside. We have about 20 to 25,000 genes. But as you saw, each of those genes could actually code for hundreds of thousands of different protein products. Potentially even millions, because at each step in that hierarchy, that cell from DNA to RNA to protein, there are levels of regulation that give different gene products. So computationally, this is just a nightmare for biologists. When we thought we understand what a gene is, how it's regulated, but when that one sequence of DNA could give up to hundreds of thousands, maybe even 1 million different protein products, that's very difficult to analyze in your head or by hand. We are now able to sequence genomes very easily. Human genome could be sequenced in a day for about $1,000 if you really have your machine running correctly. So it's very easy now to get sequence. I teach a freshman lab, and we're sequencing transcriptomes and genomes, viral and bacterial genomes, so smaller ones, with a bunch of freshmen at William and Mary. So not that it's trivial, it doesn't always work, but the point is we're generating data at this huge pace. And that figure for the 150 gigs, that is the actual finished sequence. To actually get to that sequence, you need to sequence each region to make sure it's correct 25, 50 times. So you have at least 10 times amount of that data going into the sequencing project. A lot to analyze. So what I want to do is just for a minute say that we're actually applying all of this to real biological problems. My lab is interested in how the nervous system is able to compensate, to regenerate in some ways from injury or perturbations. So we're doing this first by looking at how neural cells get their identity. You have inhibitory cells in your brain, GABAergic cells, you have excitatory, glutamatergic, and these are really important. If the balance isn't right, it leads to all kinds of neurological disease. We're interested in why certain organisms are able to regenerate and respond to a changing environment very well. And these tend to be lower vertebrates, what we call lower on the evolutionary tree. That's a very human-centric way of viewing this. But for example, fish and frogs could regenerate. They're injured, their nervous system senses that injury and compensates with new gene expression. Mammals can't do that. We have literally the same genes. You look at a frog genome and a human genome, we share, if not at the nucleotide level, but when you look at the genes themselves, the coding regions for the genes were well over 90% similar to frogs and fish. So why, when we have the same genes, can they regenerate and we can't? So we're looking at, first of all, genetic perturbations. When we overexpress a gene, you see this color that is differential. This is gene expression on one side and another. At the early stages of development, it's different, but this embryo then, by the time it's a swimming tadpole, is able to compensate, fix its nervous system. We can't do that. At the early stages of development, we're also looking at physical perturbations. We could take out from a frog embryo at the very early neural stages the entire, what will become central nervous system, rotate it 180 degrees, and these tadpoles are fine. What is happening? Which genes need to be expressed to be able to do that? That will give us some indication, hopefully, about regenerative processes. And then I don't know if any of you are familiar with Dan Crystal on campus, if any of you had him for intro bio. There's a huge team here on campus that works on mercury, looking at the effects of really low levels that you would find in tuna fish, or if you live near a coal plant, just breathing the air, or you heard about this spill down in North Carolina. Mercury is a very prevalent but low level, and the EPA says it's safe. Is it really safe? So we're looking at these environmental perturbations where, once again, lower vertebrates seem to be able to recover from damage where mammals can't. So what we want to do is not look at one gene at a time. We want to look at all the genes that are expressed in a perturbed tissue at different time points and a non-perturbed tissue to see which genes are being activated, which genes are being deactivated or repressed. And the way to do this now is RNA-seq. We use a technology called ion torrent. It's a personal genome machine. So with this torrent, they're really trying to appeal to the young generation here to say this, we just provide these torrents of data, which they actually do. The entire machine is no larger than about that, fits on a bench top, and in 2 hours, we could get 100 million bases read, 100,000 different reads for either sequencing the genome or the transcriptome. So what I'd like to do is go through some of these steps here so you have an idea of what is involved, first of all, for the wet lab biologists, where you could have some of the errors in the data and how fuzzy some of this data is that you actually get out of the instrument. And then we'll go into some of the analysis of the data and the interpretation to look at what's going on there as well. Okay, so what do? So we have this embryo, for example, that was perturbed in some way, either physically, environmentally, genetically. And by the way, this is used in disease research big time. You have a cancer cell, breast cancer cell, or lung cancer cell, and then you have a normal cell that doesn't have cancer from the same tissue. You want to compare, because most of the genes that are expressed should be similar, so you're going after the different ones. So the first thing that you need to do is isolate the RNA. We use basic biochemical techniques that you probably heard about way back in high school, where you could separate a certain type of macromolecule, just nucleic acid, and then use enzymes to completely degrade the DNA, so you're just left with RNA. That's not good enough, because it turns out that if you look at all of the RNA in the cell, and this is really annoying, that about 95% of that RNA will be what we call ribosomal RNA. And ribosomal RNA is necessary for protein translation. So we need to get rid of all of this and just go for the few percent of RNA that is messenger RNA. So if we start out with 100 milligrams of total RNA, we might end up with 2 or 3 of mRNA. So we're dealing with these totally invisible. You go blind in these experiments when you're actually doing them. You can't see anything really until almost the end. So you get the mRNA. Now you're ready to make what we call a library. So the library preparation is getting the mRNA, and mRNAs are about 2,000 bases long or so. We chop these up, so you get this mRNA, and then chop it up using an enzyme into pieces that are about 200 to 300. So everything is more or less an even size. So you fragment the RNA, and then what you do are hybridize. That's what the term means. So if you have bases that will base pair because of, again, biochemical hydrogen bonding, there are certain bases that go together, A's and T's and G's and C's, and you put these little adapters on each of the RNAs so that they all have a similar A end and a similar B end. So now all of these RNAs are chopped up with these adapters on them. The collection of those is called the library. There could be hundreds of millions of these in the library. We can't sequence that directly. So what we need to do now is something that is called template production. So we get each of these little RNAs, the millions and millions of them, and what we do is attach them to a tiny little nanoparticle, a little tiny bead. Can't see any of these. They're all, the whole tube of beads is virtually invisible that we use. But this bead will associate with the adapter on the RNA. So bead, I'll draw this a little bit bigger, nano bead here, here's the adapter, there are corresponding adapters on the bead. So hopefully you get one RNA per bead. And this is just kind of trial and error. Sometimes you'll get 2, and you could think what that is going to do to your sequencing, because you'll get double bases here. This is called, if this happens, polyclonal, because you have two different RNAs with different bases. This one could be C, G, T, and this one could be, A, G, C, and it would be trying to sequence both of those. So that's a mess. Those are thrown out. So you get your templates. So one bead, one RNA, that is now made into cDNA. So it's actually using that reverse transcriptase enzyme that I talked about as going against the central dogma earlier to make cDNA. We then amplify these, let's not have a polyclonal there, using an enzyme. You probably have heard of PCR, right, from all the crime shows? Polymerase chain reaction. So you could have the tiniest microscopic drop of blood or hair or anything on a crime scene. You can't make an identification by just looking at it or doing any other kind of test. They could get the DNA and amplify it, use primers, and then through 30 rounds, so you have one, say you have only one copy of the DNA from the suspect, you have one copy, that one in one round of replication goes to 2, in the second round to 4, to 8, to 16. You do that for 40 rounds and you end up with, math majors, you can figure that out. Lots. Enough to see on a gel, sequence, and say right away, okay, your DNA was on this victim. How did it get there? Explain it. And if you have a large enough sequence, it's virtually, and nobody messes up the crime scene and wears gloves, etc. It's an absolute identification. Absolute. Unless you have an identical twin. That complicates matters. So you use PCR to make hundreds of copies of that initial cDNA strand. So you have these little particles with hundreds and hundreds of copies. Same one now, that will get sequenced. So now finally we're able to, so here's the nanosphere, here the particle, and then here are all the copies after the polymerase chain reaction. Then it gets put into this wonderful instrument. For biologists, we're astounded because it only costs about $100,000. And that's not a lot in biology. That's really, really cheap. So we're just astounded that we could get something like this for so cheap. That's why our grants have to be big in biology. Anyway, we have one of these over in the Integrated Science Center. We've been using it consistently in our teaching classes. And what we do is take everything that we have prepared, the template positive spheres, and then put them on a chip. So this is actually a real chip that we used in our last run. This chip, you can't really see any of the holes, but it has over 1 million little holes, and those little particles go, just one could fit in one hole, and it fits in, and there are sensors on the other side, and this is where it becomes highly proprietary. They don't reveal how they do this. But there are sensors which are then attached to this instrument which sense position-specific changes in pH. During DNA replication, a hydrogen ion is released. So they claim that this is the most sensitive pH meter on the planet because it detects just one hydrogen ion. So they know that a base got added if there's this release of hydrogen ions, which is being detected by this incredibly sensitive detector. And what they do is flow the different bases over in a very pattern-known way, so you know for each position, oh, an A got added there, a G got added there, a C there. So this is how that, which is then sent to a computer which has, 15 gig, ours has now, just that within the sequencer itself, it has about a 5 terabyte hard drive to store all the data. So a huge processor so that all this information could come within 2 hours. So at the end of this then, what we get are millions and millions of bases sequenced. This is what the chip looks like. Red means all the little holes got filled with those little nanoparticles, so red is good here, blue is bad. Actually, this right here is the shape of the chip part which holds the ISPs. So this would be a great run. But then you get all this data. What was filtered out? What was a bad read? And it's not good or bad. It's this continuum, and this presents problems with data analysis. You could have data, and it's still actually being sequenced and saved, 1 mistake for every 10 bases. Of course, that would be horrendous. Nobody would keep that. 1 mistake per 100 bases. Still not good enough for any sequencing biologist. 1 per 1,000. 10,000. So you have all of these differences. What do you accept? So that's part of the issue, and different platforms will have different cutoffs. So you have all these filters. You're going to get some polyclonals where you get 2 bases instead of 1. We don't have a good way of separating that out, even though the sequence was good. Then you get to the point of, all right, you have your 100 million bases, say sequencing all of the RNA that was present in a given cell, the cell that was exposed to mercury as opposed to a cell that wasn't. Now what? What do you do with this? This is the real problem, at least for biologists, and actually for the field in general. This kind of falls into just the data analysis, not even interpretation yet, 4 different basic components here. So just to give you an idea of where we're going to go with this, what you need to do here is somehow get all of these little reads. Remember, you don't have the whole transcript because you cut it up into those pieces that I was talking about earlier. Different sequencing technologies have different length reads. The most popular one on the planet has reads of 20, 30, 40 bases. This is nothing. How do you reassemble and make sense out of them? Ion Torrent has reads of about 200. Now everybody's trying to extend these, but you never have the whole RNA. So actually aligning these back to a genome and calculating and normalizing, because every run's going to be different, the reads per kilobase of gene per million reads altogether is the way we generally do this. But this has been hugely criticized in the literature as a way to normalize. So you need to do something like this. You want to find out what transcripts are there, and then you want to find out if there are any mutations in the RNA that are giving rise to aberrant proteins. So you want